large complex molecules responsible for the sustainability of cellular life. The basic units of proteins are amino acids bonded together through spontaneous or intermolecular forces, which form peptide bonds between the amino acids, forming a polypeptide. Polypeptide is an organic polymer consisting of one or more amino acids. This polypeptide will fold into a specific 3D structure to form a protein. Proteins are embedded into every aspect of a living organism's life, from food to bones, muscles, and even hair. Proteins aid multiple essential functions for cells, such as providing structural components for our tissue, as well as antibodies, enzymes, messengers and hormones, transport, storage, and contractile movements. Without these driving proteins, our cells would cease to perform basic functions, thus would lead to the decline of the cell's life. Polypeptide synthesis is the biological production of proteins within cells to balance and replace pre-existing cellular proteins, occurring in two steps: transcription and translation. Transcription is the first step in the production of polypeptides. It is the process of building a chain of polypeptides from amino acids. Polypeptide synthesis begins at the nucleus of a eukaryotic cell. The double-stranded DNA molecule will begin to unzip and unwind as the helicase proceeds through the two double strands. The new open region is now called the transcription bubble. This action allows RNA polymerase to bind to one side of the DNA molecule, forming the template strand. This initial process is called initiation. RNA polymerase is an essential enzyme that binds the free nucleotides together. The RNA product will be complementary to the template strand and almost identical to the other original strand, thus making it the coding strand, with the exception of uracil on the RNA molecule instead of thymine. To help transcribe information, RNA polymerase builds a new RNA molecule using the template strand by collecting free nucleotides from the nucleus and pairing them through the base complementary pairing rule. For instance, if there is a T on the DNA template, RNA polymerase will help bind an A to the new growing RNA strand. This RNA strand will continue to grow and add new nucleotides from 5' to 3'. The completed RNA strand will become messenger RNA or mRNA. It's responsible for transferring the genetic information to the ribosomes for translation. mRNA is key to transcribing the correct information, otherwise defects can occur leading to the faulty production of proteins, which will not establish and execute its job correctly, leading to cell death. Each protein needs a specific arrangement of RNA molecules in the mRNA to become fully functional for its purpose, thus start and stop codons are needed to accomplish this task. A codon is a set of 3 nucleotides with different purposes. One that will signal to the cell when to first begin sequencing the nucleotides and when to stop bonding new ones. There are various combinations of start codons, but the standard one is AUG. Similarly, there is a range of stop codons consisting from UAA, UAG, and UGA. Finally, termination will occur as the RNA molecule will leave the nucleus via complex nuclear pores. These pores are small enough in size, thus doesn't allow DNA to leave as its molecule structure is too large, whereas single-stranded mRNA is small enough to move through it. As it transitions out of the nucleus, it aims to find and attach to a ribosome located in the cytoplasm. Ribosomes are cellular structures which assist in the production of polypeptides composed of ribosomal RNA or rRNA, responsible for the function and structure and ultimately the home of translation. They consist of two subunits, large and small, which allow mRNA to feed through. Once that ribosome mRNA structure assembles, the ribosome provides a template for tRNA to find matching codons on the mRNA template to ultimately deliver amino acids. This structure has slots called A, P, and E sites. tRNA or transfer RNA are single-stranded molecules with some bonded sections through complementary base pairing, giving a clover-shaped structure. On the bottom of the tRNA molecule is an area containing anticodons, a set of 3 bases, which is complementary to the mRNA codons. The anticodons are the correspondence to a specific amino acid attached to the tRNA molecule. They are exchanged into the ribosome with a mRNA molecule to proceed with translation. tRNA molecules can be found within the cytoplasm and ribosomes. Translation is the process of building a polypeptide chain from amino acids. mRNA grouped into codons of 3 bases will feed through the ribosome. A tRNA molecule will insert itself into the ribosome and bind to the mRNA codons. The first matching of codons usually produce the specific starting amino acid from the anticodon AUG. This first step is called initiation, forming an initiation complex. Elongation will subsequently occur as amino acids are individually placed by tRNA molecules moving through the ribosome along the mRNA. After the start codon is matched into the P site of a ribosome, another matching tRNA will land in the A site, causing the formation of a peptide bond connecting 2 amino acids, essentially producing a small polypeptide chain. Once the bond is formed, the mRNA is pulled through the ribosome by 1 codon at a time, causing the first tRNA molecule to exit through the E site, now exposing a new available codon slot on the A site for another tRNA, ensuring the cycle continues. Elongation requires sufficient energy to proceed. The peptide chain will continue to grow as needed until termination takes place. Termination is the final stage of translation. As the ribosome reaches a stop codon, termination happens when a stop codon in the mRNA, usually UAA, UAG, or UGA, enters the A site. To recognize stop codons, proteins called release factors fit nicely into the P site, even though they aren't tRNA molecules. They act by binding to the stop codon, affecting the ability to form peptide bonds, then hydrolyzing, or adding water to the molecule to the last amino acid of the completed polypeptide. This ultimately separates the tRNA and chain, causing the release of a polypeptide. Once the polypeptide is released into the cytoplasm completely, it will spontaneously fold into a specific arrangement depending on the order and the bonds of the amino acids. This specific fault is fundamental to the activity and the role of proteins. For example, an enzyme has a specific shaped active site due to the arrangement of amino acids, so the substrate complex can effectively bind to the active site. When the active site is disrupted and changed on the protein, its vital role can't be performed as the substrate won't fit the active site.